Okay, great. Thanks, Brendan. So let's move on to the first talk. So this is Mike MacCoss, who's a professor in the Department of Genome Sciences in the University of Washington, is also a long-time quantitative proteomics guru, and in particular, I think his group has really been pushing the method development and software development in the DIA space. So we're very happy to have Mike here to give us his version.
Great, thanks. Yeah, so wow, that's fantastic to see everybody here at this first DIA SWATH course. This is fantastic. It's great to see, ideas that have been going around for a long time finally maturing to a state where we actually have a course on this very specific to this. I do actually. I'm trying to think it's right here. That's probably what's causing. It's in the same pocket too. So it's absolutely fantastic to see things kind of maturing this way. I hope people realize that this is definitely the first course that they've done with this, and I'm sure everybody's going to love some different feedback. So one of the things that I'd like to talk about is kind of a little bit about how we kind of view data-independent acquisition. And in our lab specifically, I think what will be a little bit different is we're going to talk a little bit about some of the specifics about doing this on Orbitraps, and also a little bit about the topic of peptide-centric versus spectrum-centric analyses in general. And so I'll point out that I'd much rather people just stop me, interrupt me, and have a discussion if we want to. If we need to kind of move some stuff to Thursday, I definitely am willing to do that. And I think it's more important to have people kind of up to speed and kind of understanding what we're thinking about. I'll also agree that I'll also point out that not everybody agrees with some of the things I'm going to present here. It is kind of a very new field, and so I think that's kind of an important aspect of this course, is that it is kind of at the early stages of things, and it hasn't really matured into kind of consensus for how to handle things. Okay, so just to get started, make sure we're kind of all on the same page. This is the way we kind of view kind of the proteomics data acquisition strategies. So there's data-dependent acquisition, which basically we sample things based in real time, instrument making real-time decisions on the fly. And we generally have narrow precursor windows. We get relatively clean product ion spectra. We do then database searching and have some sort of peptide identification step. On the other extreme to that method, we have like parallel reaction monitoring or selective reaction monitoring. And in this case, we actually tell the mass spectrometer to measure very specific precursors because we're interested in specific peptides. And as was already brought up earlier this morning, you basically sample these things in a systematic way. So we're repeating the acquisition of MS spectra in a cycle time that is reflective that we can then reconstruct the chromatographic peaks. We then extract the product ions from these data. And this is just an example showing parallel reaction monitoring. And what the hope is, I think all of our hope is that data-independent acquisition allows us to kind of bridge this gap, right? So here we can kind of sample lots and lots and lots of things, thousands of peptides, sometimes tens of thousands of peptides. In this case, we're basically measuring hundreds of peptides. And we have to have preconceived notions of what we want to be measuring. And in this case, what we want to do is cover a relatively broad range that's fairly comprehensive, but it's not completely comprehensive. It's in a relatively reproducible cycle time. But in order to cover that breadth of the range, we obviously have to open up the isolation window and make it so that we essentially are multiplexing the MS acquisition by intentionally sampling multiple things at the same time. And this then results in spectra that are very complex. They have lots of components in. And the chromatograms, while some transitions tend to be fairly interference-free, some tend to have some significant interference. So this is basically where we're at, right? So we want to make sure we collect data that's systematic, that we can then reconstruct chromatograms, because we find that there's a value in integrating a peak area. And we feel like there's also value in doing the quantitation and detection on the MS2 level. But at the same time, we need to somehow deal with this decrease in selectivity. So just to make sure people are all on the same page, one of the problems we have in this field is that everybody's kind of given this a different name. So data-independent acquisition was originally a name that John Venable, who was actually a postdoc in John Yates' lab, kind of coined it. And his intent was to kind of say it's the opposite of data-dependent acquisition. It's data-independent acquisition. He wasn't selecting precursors based on an MS1 scan. Jim Bruce's lab at the University of Washington also called this all reaction monitoring. Of course, it's got other names, SWATH and HRM. And I think, the way to kind of describe this is basically kind of like Kleenex and tissue, or and so they both mean the same thing. Some of them actually are trademark names, some of them are not. But they do add a little bit of confusion to the field, and they all have slightly different nuances. So sometimes people say, SWATH can only represent things that are being done on SCIEX, QTOF instruments, etc. So I personally like the name data independent acquisition, largely because it's a little bit more general and broad. So I'll get to a little bit to how we kind of came about this before, but the simplest method that we would run on an Orbitrap type instrument is a 20 by 20 mass of charge wide window experiment. This covers in general about 400 mass of charge. And we then measure this from 500 to 900. So we're basically measuring all fragments for all precursors between 500 and 900. And I'll get into a little bit more why we have selected this region. And especially on Thursday, we'll talk about advances for how we're trying to solve this 20 mass of charge wide window problem. Data independent acquisition has been done on basically every possible mass analyzer. It was originally done on linear ion traps, and I actually believe kind of the future will come back to linear ion traps. We'll talk a little bit about that on Thursday as well. We'll be talking predominantly on Orbitrap instruments, but there's been papers reporting it on time of flights, obviously, and then also FTICRs. In fact, Jim Bruce's lab has done a lot with data independent acquisition on FTICRs. One of our colleagues Jake Jaffe likes to call it comprehensive MS-MS acquisition. I kind of like that term as well. It's not comprehensive proteomics, but it is comprehensive MS-MS acquisition because we are sampling everything comprehensively between a given mass range. And the way, of course, this works, in one scan, we collect one MS-MS window, another one we do another window. Until scan 21, we repeat the process again. Another kind of critical component to this is that we need to make sure that this is occurring in a frequency that enables us to basically collect data across a chromatographic peak. So if you have a 30-second wide chromatographic peak, you're basically then sampling it 15 times across the peak. Again, on an Orbitrap, in most cases, we're doing a quadrupole Orbitrap experiment. For a QQ Orbitrap type of hybrid instrument. So this can either be a Q exactive or a fusion or a Lumos. And if you look at a given isolation window, we're passing intentionally multiple fragment ions at once. We're subjecting those multiple fragment ions to collisions, and then we're measuring that mixed spectrum simultaneously. And we do throw in the MS1 scan basically because we get them for free. The instrument does actually works is really fast to acquire MS1 scans. It's a little slower to collect MS2 scans, and a lot of that comes down to just the time it takes to fill the Orbitrap. And again, we'll talk a lot more about that later. So just a little bit of review here. So data independent acquisition in most cases has comprehensive MS1 and MS2 sampling across a given mass range. For any one given isolation window, it's untargeted because the center of this isolation window, in this case it's placed at 510 mass of charge, is not directly centering it on a given precursor. So in this case, the precursor could be on one side, it could be on the other, etc. It's also unbiased because we don't care what the intensity of that precursor is. One of the hallmarks currently, and this is something I believe our field has to change, and that is currently these methods are known for having wide isolation windows, and in this case I'm illustrating at 500 to 520, and it's also collected systematically across time. So it's a consistent or systematic sampling, and we do this on a chromatographic time scale. Now, for me, one of the things that kind of drew me to collecting data by data independent acquisition, because we had been collecting data independent acquisition for a very long time, going back to around about 2004, 2005. And one of the things that was a little different was once we started working with Skyline, we started thinking, you know what, I wonder if we could just go and pull out some of these individual peptides out of these data sets. And Brendan had made it so that Skyline could, in theory, do that. And so if you had kind of, say, a fairly wide mass range and you had this specific peptide that you're interested in, the doubly charged precursor was at 790.4, you could calculate which window it should fall into. You could look at this individual window across all of the scans across time. You could then predict the B and Y fragment ions, and you could go into each individual scan and extract that information. I'll point out here that this is actually a very clean piece of data. So you basically have mass spectra collected systematically and continuously across time, across retention time in this dimension. And then at each individual mass range, we have different fragment ions that represent, of course, the sequence of this individual target peptide. And in this case, this peptide happens to be the most abundant thing in the isolation window. It's rare that this is the case, of course. Most of the times, things are 10 or 100 times less abundant than the most of things in the isolation window. But when you look at the data, it looks very similar to what we get with selective reaction monitoring or parallel reaction monitoring. So this, again, is something that we've definitely taken very closely to heart. We think that we like doing targeted experiments on relatively finite numbers of things. And we feel that data-independent acquisition simplifies the data acquisition process, and that's a good thing. It does, though, complicate a little bit of the analysis process. I'll talk a little bit more about that later. So in many ways, one way to think about this, and this is something we've kind of thought a fair amount about, is you can think about this as being kind of PRM or SRM at all. So in a case of parallel reaction monitoring or selective reaction monitoring, you may have individual target peptides, which you can go in and extract chromatograms for each individual peptide that you're interested in. And of course, there is a finite number of windows that are measured at any one point in time. But in the context of DIA, in theory, anything that falls in the isolation window is game. And you can, in theory, extract chromatograms for any target peptide, precursor, and product ion data. I do really want to kind of emphasize this importance of chromatograms. We're so used to thinking about things as spectra that I really do believe that one of the key things about data-independent acquisition is the fact it's a chromatographic method, that we look at our data as chromatograms often, and the signals that we represent are some sort of integrated signal across time. That's the reason why we got drawn to it, because it's really hard to do quantitative analysis off of individual spectra, and it's much easier to do quantitative analysis off of chromatograms. But this does require people to be good chromatographers. Good chromatographic peak shape is a really kind of key component of this. And this is something that people will get often good DDA results with good spectrum counts, and then they're not quite clear why they're struggling with their SRM or targeted methods. And it's largely because data-independent acquisition is a targeted method, at least from my perspective. So there are some differences, of course, between DIA and SRM. There are more than just, let's just kind of review some of them. One is the isolation window, and this is really kind of the key component. So I've talked about the simplest experiment as being kind of the 20 mass charge wide isolation window. In a triple quad, we almost always use unit resolution, or 0.7 mass charge as the precursor window. The second kind of key difference is one is untargeted and one's targeted. So one, the isolation window can fall, the target peptide can fall anywhere in the isolation window. In the case of targeted, you actually set the window where you're at targeted mass of interest. In one case, it's narrow. In the other case, it's wide. And then when you look at the mass analysis, in the targeted analysis, one uses a mass filter and one uses a full scan. So in the case of this, it's unit resolution, but we measure individual masses one at a time. We don't measure a full spectrum at a time. So we set our quad to measure one mass, and then we set it to measure another mass, and then a third mass, etc. And these all have to be defined in advance. But basically, we use this information to redefine a chromatogram. In the case of a DIA experiment, we actually do a full scan. So we collect the entire spectrum at once, and then we extract the masses of interest. But in the end, the data looks basically the same, and we use very similar tools to integrate and determine the quantities. Okay, so what are some of the advantages of data-independent acquisition? So this depends on who you're talking to. But from our perspective, one of the things that's really nice about this and appealing is that the first thing that you often start with in the case of an SRM experiment is you have to define what is it that you're going to measure to start with. So you'll go through saying, look, we're really interested in X protein. We're going to use, these Y peptides that we're going to use. We're going to go about retention time scheduling, transition selection, refinement. And then you finally get down to running your samples on those finite sets of peptides. You then go through peak integration, and then those, of course, any experiment generates, either confirms your hypothesis or generates new ones. In the case of data-independent acquisition, in theory, you collect your data. You generate a hypothesis. You look for a set of peptides in your data, and you go through this chromatogram extraction, peak integration, transition, refinement, and then you generate new hypotheses, but you go back and reanalyze the data again. So this is particularly valuable, right? Because one of the reasons we got into this is we would have a huge collaboration with somebody, measure a set of peptides, build a set of peptides to measure 100 peptides, say, in a set of samples. You'd measure that set of samples on, say, 100 different samples. And then the first thing that their collaborators would say is there's a brand new protein that someone said may be important in this case. Can you go back and rerun those samples again, measuring this protein? So what is kind of nice is that in theory, you can collect the data once, and you can go back and actually have your collaborators, in theory, reanalyze the data themselves with some training or knowledge about this. In practice, it tends to be a little complicated to kind of interpret these data. So these are something that that everyone's still working on to make easier. But still, I think this is basically the reason why people like this. In fact, it tends to be very appealing for some, like core labs in particular, because there, in theory, you could collect data once on a very valuable set of samples, and then use that in order to be able to go back and have people ask new questions over time. So one of the promises of data-independent acquisition, and that is that you collect this molecular image or digital archive of the sample, you mine it over and over again. There's no scheduling, which tends to be one of the complicating factors of doing a targeted SRM or PRM experiment. You can make direct queries, so you can basically say, do I have evidence of this peptide? And I'll talk about this in a second. You get better quantitation than you do with any MS1-based quant. So it's really kind of, while it's not as good as using a narrow isolation window, it's definitely much better than using discovery data. And I've shown here on the right-hand side an image of the Mona Lisa, because one of the analogies I want to draw here is similar to what Google has this project where they've generated these gigapixel cameras. And their goal here is to kind of generate a permanent record or a permanent archive on famous works of art, right? And famous events and famous people. And they didn't want to, of course, take an image that was going to be part of the permanent record using basically your cell phone camera. They wanted to have something that had such extreme number of pixels that it was basically going to be a permanent record. So if you think about it this way, acquire a molecular image or digital archive of the sample. In general, you'd want to have this where you collect the data with the highest resolution possible. So let's kind of look at this a little bit, right? So here's a photograph that Google took of the inauguration. And if you look at it closely and you blow up certain portions of it, you can actually get these incredible blow-up images of certain individuals and how happy or unhappy they may be. And so one analogy I kind of like to give is we'd like to kind of think of this as being, as a DIA is the equivalent of kind of like the gigapixel image. You're collecting this really detailed amount of information across an entire image or a broad range, where the equivalent of parallel reaction monitoring or SRM would be like the equivalent of collecting the data just on very specific individuals. So you can have a telephoto lens, get an incredibly high quality image of a sub portion of that total image, but you're missing the bigger picture in many ways. You're looking at it a little bit out of context of everything else. So, of course, sometimes we know that there can be valuable in keeping the full picture, just in case you need to go back and to have extra evidence to show who had more people attending an inauguration. You actually have that data. Of course, you can show then kind of the full picture. Okay. So there can be value in keeping the full picture, and a couple of things that our lab has done. We've had collaborations before where people have said, look, can we use different peptides to measure this protein because of some new splice variant that somebody's reported. We've gone and looked for different proteoforms. We've looked for a specific oxidized residue that somebody had said was going to be important in a given sample. In some cases, we've gone back and we've chosen different transitions. A peptide had interference in the data, and we said, look, we want to drop that one, and we're going to add another one. So this allows us to do that because basically we have all the data. So one thing that's also I mentioned previous, the reason why this is particularly powerful is that on the left-hand side, that's what we did. We took 30 bovine peptides, we spiked it into a complex matrix, and we did an initial DIA experiment. And one of the things we wanted to do was to kind of compare the quantitative lower limit of quant. So we did dilution curves in this background. And if we use data-independent acquisition, in most cases, it's about between like 4 or 5-fold increase in lower limit of quant, depending on the method. And I'll point out, with the exception of when you're using a very large isolation window, you tend to then lose some of our quantitative ability, and that's largely because there tends to be a lot of interference in the MS2 peaks, that when you have a large isolation window, that the lower limit of quant in a 20 master charge wide window may not be that much better than MS1, but when you use something like a 10 master charge wide isolation window, and we'll talk about this on Thursday, these overlapping windows, which is effectively a 10 master charge wide window, we tend to have a lower limit of quant that's substantially better than that of using an MS1 data. So how do we think about analyzing this data? I know that you've talked a little bit about this in the last day or so. I kind of want to give an analogy. So one of the reasons why we like data-independent acquisition is because it really is kind of a very targeted way of looking at your data. So let's think about the fact that this is an analogy that Jared Egerton likes to give. Let's say you're going to the library and you want to find your favorite book. And so you have a specific book that you're interested in, and let's say it's Harry Potter. And so let's say you're going to do this by data-dependent acquisition. The analogy for data-dependent acquisition would be is that you told the librarian to just go randomly pick out books in the library, and you hope that you happen to select it, right? So then you go and you select it, and you say, these are the books that were on this cart that the librarian pulled out, and none of them happened to be the book that you're interested in. So you can be pretty happy because in this process you can say, look, there's these other books I might possibly want to read, and so these are the ones I'm kind of forced to deal with, but I can't really say anything about whether or not the library even had that book or whether or not I could have gone and found it. So kind of the conclusion is that Harry Potter was not identified, so you could incorrectly kind of assume that the library does not have it, but this could be just an artifact of the sampling. So another way to think about this is kind of a targeted analysis, and that is that we know a lot about this book. We know, let's say it was sorted in the library alphabetically, so then you could go and say, look, I'm going to go and look for these things, this book under H. Things like the size of the book, what the book cover looks like, things like the author. And this is basically the way we want to treat the DIA data, is we want to start with things like we happen to know the retention time of this peptide. We know which peptides are stable. We know which ones have a good, which product ions, what are the relative fragment ions intensities. So we know a lot about the information of an individual peptide. We know basically where to go in the library to look it up to begin with. So Sonia Ting had kind of tried to summarize this. We had a perspective paper that was a couple of years ago now, and she tried to get this across. I still think it's maybe confused things still, if nothing else, but at least it's been a starting point to think about this. So the way she was trying to say it is if you have spectrum and you're trying to query them against a sequence, and then in there you have resulting scores of peptide spectrum matches, of which then you compute p-values and q-values. If your query unit was spectra, so you're starting with some sort of data looking at sequences, that's probably a good way to call this is a kind of a spectrum-centric analysis. An alternative, and one thing that we've been kind of advocating for, and other people have been advocating to as well, and that is this idea of a peptide-centric analysis. And this was, of course, first reported in the Gillette paper, and you have this peptide-centric analysis, where the hypothesis is a little bit different, and that is, do I have evidence basically for my peptide in the data? So you start with an individual peptide of interest, you try to extract some information about it, you compute some different scores, and in this case now you compute p-values and q-values for this individual peptide, when in this case you have a p-value, q-value for the peptide spectrum match. So the information is a little bit different. So in this case, you get basically a direct p-value, which is something that we're basically interested in, right? So one of the things, the hallmarks of analytical chemistry, anyone who's taken any basic quantitative analysis course knows that one of the things that you learn early on is how do you define your limit of quant and your limit of detection. And in many ways, I think this peptide-centric analysis and the p-value is basically whether or not you have evidence for that peptide. It's whether or not your peptide is above your limit of detection. So this, I think, that's the closest analogy that kind of brings it home to kind of a lot of kind of classical analytical quantitative analyses. So I'll point out that when we first started doing this, I thought that we would just be able to go and look at these data like SRM data files, but the peak picking is definitely a challenge. So if this is basically an individual target peptide, and here's the MS1 data, and here's the MS2 data, this region right here is where the actual target peptide is, and it's pretty messy, right? The data is very noisy. There's lots of other things that are interfering with it. The MS1 data is completely confounded by other things at that mass, and that you can't really distinguish it from the background at all. The MS2 data, in this case, is noisy, but there are definitely transitions that allow you to integrate the area under the curve. But just picking this signal, this retention time out of the data without a standard would be pretty hard. But we actually happen to know a lot about these peptides. We know the retention time, we know the fragment ions, et cetera. So there are, and I'll also point out that we're in kind of a very interesting time. There started off to be only a few tools initially to kind of get going, and that is, of course, things like Open SWATH, PeakView, Encyclopedia, or Spectronaut, and Skyline, of course. And then I'll talk a little bit more about Skyline in a second. And then with no spectrum library, there's now 2 tools out. We have a paper on Pecan, which I believe is accepted. We should find out literally any day now. And then the FT-ARM is a method that Jim Bruce had kind of published. And then there's these other tools which kind of still analyze DIA data, but they take it in the context of a spectrum-centric result. And that is they try to demultiplex these complex spectra and try to still treat it like a search result. You're still, in essence, getting a peptide spectrum match, and you're not necessarily getting results for every peptide that you query against your data. So this DIA analysis in practice, again, is kind of a mess. You basically start with a bunch of your precursor chromatograms, and you then take your bunch of your product dyne chromatograms. You extract the data, and you need to basically be able to say whether or not you have evidence for the individual peptide, and if so, what is the retention time bounds for which you're going to integrate that peak in order to arrive your signal from your quantitative data analysis. So the most common way that this is done is using what people call a spectrum library search workflow. And I think you've already been introduced to this a little bit. And I know you'll be introduced to it a lot more. And that is basically you have your wide window DIA file. You have a spectrum library which contains information like what are the relative intensities of the individual product dynes, but also your IRTs, your retention time information. You then compute different scores between this library and the wide DIA file, and then you use a machine learning classifier to basically help try to distinguish your correct from incorrect results. This is implemented in Skyline. This is a tool that basically that's all derived off of mProphet that was developed in the Aebersold lab. And of course, there are lots of tools now. In fact, all of these tools essentially are derived off of the original mProphet algorithm. And it's not that surprising that almost all of them give very similar results. And so that's one thing that's kind of an important thing to kind of keep in mind. These are all based off of a very similar sort of machine learning approach, uses very similar set of features. And while they all have slightly different spins on them to make them either more comprehensive or easier to use, in general, they all perform fairly similarly. So there is a couple things, and that is that in order to then have that spectrum library, you then need to do some data-dependent acquisition runs or collect spectral libraries beforehand. There's still, in our case, when you look at these data in Skyline across a large number of runs, you'll notice that almost all of these tools still make mistakes. And being able to visualize these data and to be able to correct them manually is very important. It does, though, require a fair amount of labor to do that. And this is one of the things our lab struggles with the most, is still trying to go through and feel like they're comfortable with the results that are being output. And the other thing is that I'll talk about this in the next few minutes, is that you're kind of limited to only things that have been seen previously by data-dependent acquisition. We already kind of talked a little bit about the fact that data-dependent acquisition may not catch everything, and so if you're starting with that as your starting point, that's kind of maybe not necessarily the best place to start. The other thing I'll point out too is that the MS2 is way more sensitive than the MS1, but with data-dependent acquisition, we're triggering off the MS1. And so we're basically ultimately going to be limited by whether or not we can distinguish the MS1 signal from background, which is definitely a challenging point to start with. So often that will require doing things like fractionation or doing other tricks in order to improve this. Okay. So one of the things that we talk a lot about in our lab is that everything comes down to DIA being about balance and sometimes sacrifice. So there are lots of different things that we want to try to optimize, and unfortunately, to improve one set of parameters, we actually are sacrificing the other. So the first one I'll point out is the mass to charge range covered. In our lab, we almost always collect our data from 500 to 900. This is a much smaller mass to charge range than what other labs do. And we feel fairly comfortable with it, though. And the reason for this is this experiment that was done, I think it was originally done in about 2009, but didn't get published until about 2014. And that was, we took basically 2 mass to charge wide isolation windows and measured it through multiple different injections across the entire mass range. So this is an experiment that Dave Goodlet's lab has called Pacific, and it's actually a very powerful experiment. And our lab, we'll talk about it on Thursday, has kind of adopted this strategy for building all of our libraries now in-house. It just takes a few injections, but it actually makes an enormous difference at defining the things that can be detectable in a set of samples. The key thing here, though, is that most of the peptides found are in this 500 to 900 range. It would be nice to be able to measure from 500 to 1,000, but in general, 500 to 900 is pretty good. And by extending to include wider windows, we either have to decrease the sampling across a chromatographic peak, or we have to increase the isolation windows. Both of them actually hurt our analyses more so than the narrow width at which we're sampling. So in our case, this tends to be the starting point for almost all of our data. There is a couple people in the lab that do 24 mass to charge wide isolation windows, but they do a trick that gives them 12 mass to charge of specificity back in the end. I'll talk about that on Thursday. So again, if we start with this 20 mass charge wide windows, and if the instrument can scan at kind of a fixed amount, so let's say 10 hertz. By 10 hertz, I mean 10 spectra a second. And your chromatographic peak is 30 seconds retention time. You're basically getting about 15 points across the peak. You could narrow the isolation window and do 40 scans to cover the same mass range, but that would mean that you're sampling the peak, chromatographic peak, about half the width, half as frequently. You can go even smaller, but then you're getting to the point where you probably don't want to use this data for quantitative analysis. And so the main reason why you wouldn't want to use this for quantitative analysis is because you're taking the signal here, 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 and here to define your peak, and you're going to be significantly underrepresenting the area of the peak. So in general, you need 8 to 10 points across the peak in order to be able to accurately define the area. And so in general, that limits us basically to doing things either 20 mass charge wide windows or 40 10 mass charge wide windows. This is pushing it, though. One thing that's a kind of a trick, and don't rule this out, in fact, this is something that our lab does a lot of, is making multiple injections. So you can make 2 injections of, which covers 40 by 5 mass charge wide windows. And this kind of gets you back up to the same sort of sampling frequency of the 10 mass charge wide windows. And so in general, we have found that this is a very useful strategy is to make multiple injections to cover a given sample. There is a negative downside to this, and that is that instrument time is money. You're basically depreciating the instrument cost per unit time. And so if you have to double the number of analyses per every sample that comes in, you're basically doubling the cost, right? So this tends to result in a little bit of a challenge. So if you think about things as being DDA tends to be these narrow isolation windows. Now people are doing much smaller windows versus DIA with different isolation widths. The lower precursor selectivity for DIA is definitely a problem. More peptides are co-fragmented. Most cases, most isolation windows have multiple things in them. More complex MSM spectra and more interference. Just to kind of illustrate that again, so here's looking at a spectrum with 2 mass charge wide isolation window, 10 mass to charge, 20 mass to charge. You look at the chromatograms, 10 mass to charge, here's the chromatograms. You see a little bit of interference in this one transition. But once you go up to 20 mass to charge with windows, of course, there's a whole other peak that shows up on the side. And one is basically on the outside of this right-hand side of the mass than the other. So this is kind of the thing that's really kind of hit home for us. And that is when we started doing experiments, these are data collected only from 500 to 700, so really narrow total mass range. And we did the experiment with varying different isolation widths. You'll notice these are all very small compared to what people are generally talking about, DIA or SWATH. And we did, in some cases, we did 1 injection, but we didn't want to go beyond a 4-second cycle time. But in general, the cycle time didn't affect our ability to detect a peptide. It affected our number of things that we could detect dramatically. There's almost a linear response, an inverse linear response dependent on the isolation width. So if that means if if you're going, here is about 10 mass to charge, and you're looking at around about 8,000 or so peptides. If you go down to 5, it's around about 13 or so. So it is a pretty significant increase in the number of things you can detect in a given isolation window, depending on the isolation width. So in our mind, this is the single biggest thing that we need to improve upon. In fact, we've been kind of largely on a goal to get down to this 2 to 4 mass to charge wide isolation width. We'll talk a little bit about this on Thursday, and there's lots of kind of tricks that we've done to try to get that while still spanning a wide range. So people often ask, well, why is it that that we're able to get more peptides per detection when we narrow the isolation windows? It largely comes from the discriminant score. So if we have a score that we're calculating for those that are present versus those that are absent, normally, of course, we set some given false discovery rate. In this case, it's a really restrictive, it's 0.1% false discovery rate. And if you narrow the isolation window, you either shift the peptides that are present or the ones that are absent that are further away from one another. And this then allows you, of course, to draw your threshold line a little bit more to left as well. And this increases the number of peptides you're able to detect. So the better you're able to distinguish between those that are correct from those that are incorrect, the more things you can detect. So, and we're not the only people who have kind of said this. The precursor mass of charge is probably the most important thing. So here's this one figure from Bruno Demond's lab in JPR 2013. This is definitely a great paper to read if people haven't seen it before. And basically they plot the product dyne resolution versus the precursor resolution. And it is kind of an interesting thing. If you actually read the figure legend, one of the things that they point out is that the product dyne resolution is very important. But I would actually point out that the precursor resolutions, their data and our data show that the precursor resolution is more important than the product ion resolution. So while, yes, there is a slight decrease in your or in the amount of interference when you go to a higher resolution on the product ion resolution, there is a much, much greater decrease in your fragment ion interference when you decrease the precursor resolution. So the argument that I'm going to make here is if you are going to basically come up with this permanent archive of your data, what I'm pointing out is we're basically at kind of generating these permanent records like we are, like this right now. If you narrow your windows, you can do it over a narrower mass range, but you're going to increase the resolution, but not on the entire image. And of course, you could then increase the number of injections to basically get a little bit better of a resolution. So I want to talk a little bit now on some of the things that are very specific to Orbitraps. How many people here are thinking about doing DIA on Orbitrap mass spectrometers? Wow, so it's easily more than half the room or half the room. So that's good. So one of the things that wasn't quite obvious to me when we started doing this was what Thermo was advertising as being the scan rate. And so this is a figure that we made, and it's based off of a QEHF. And so Thermo will tell you that the QEHF can scan at 20 hertz. So it's 20 spectra a second right here. Now the thing is here, is what we've shown here is that if you're scanning at a given scan rate, what is the maximum injection time that you can use to fill the trap? Yes, the Orbitrap can basically go at infinitely fast speed. It just decreases resolution. So this is at, this black line right here is basically showing things at 15,000 resolving power. This is showing 30,000 resolving power, and these are showing 60,000 resolving power. So as you have a longer resolving power, you actually have more time to basically fill the Orbitrap because you're basically scanning slower. So one of the key things that we point out to people is that you really don't want to have much less than 60 milliseconds to fill the Orbitrap. And so that's basically looking at about 10 hertz. And so if you have less than about 60 milliseconds, you're worried you're going to run into the chance of potentially going and underfilling the Orbitrap, which means you're going to sacrifice sensitivity. So yes, you can go at 20 hertz. Whoops. But this then means that you're limited to about 17 milliseconds as the maximum inject time. Now, why is this important? Because this is the amount of time that that if you're scanning the mass analyzer, you're not scanning it, you're actually just you're measuring the image current for about at a rate that allows you to collect spectra at 21 hertz. It means then the most amount of time that you can actually spend filling that Orbitrap is 17 milliseconds. So if you have a particularly weak signal, you want to spend a much greater period of time filling that Orbitrap than if you have a more intense signal. So just to kind of illustrate this a slightly different way, this is a kind of experiment done using 40 by 10 mass charge method Underfills. And so here is basically the number of ions that are measured in each spectrum versus the number that are measured when we do 20 mass charge wide windows. And I'll talk again a little bit more about the overlap on Thursday. But in this case, this is 17 milliseconds maximum inject time to scan at 20 hertz. And this is the case where we're basically scanning 10 hertz. And we were putting in actually a much, much greater number of ions into each spectrum that we're able to use for quantitation. Another thing that I'll point out is that not only do wide windows affect the qualitative ability to detect a peptide, it does affect our ability to actually measure the quantity of different peptides too. So here's 150 peptides that are spiked in at 1 and 2 femtomoles into a human neuronal cell background. And these are all the peptides that were measured, and then green is basically parallel reaction monitoring. Notice that there is kind of a fairly narrow kind of distribution around the center of the peak. As you get wider and wider windows, the distribution gets more diffuse. Also, the precision gets slightly worse too as you get to the point of wider and wider windows. So one of the things that we decided to do when we first got when we first started working with Thermo on the Orbitrap Lumos was to kind of assess a bunch of the different method capabilities for data independent acquisition. And the first thing that we did was basically we have, of course, data independent acquisition where we have these wide isolation widths, we have the chromatographic extraction, and we wanted to evaluate things like the precursor selectivity and cycle time, fragmentation method, and product ion sensitivity. So I'll talk about these first two ones first. So one of the things that's a little unique about the Lumos or the fusion instrument is that we can do two different types of activation. At minimum, two different types, but there's two different types of CID resolution. One is a beam type fragmentation, and the other one is resonance CID in the linear ion trap. And so they both have slightly different pros and cons. So in one case, the activation speed for resonance CID is slower than that for beam CID. The number of the fragmentation is going to be different. So in resonance CID, as soon as you break one bond, it's off resonance, so you don't get any more secondary fragmentation, or you get very little secondary fragmentation. When beam CID, you pass it through a gas, and you'll get multiple collisions. There's resonance CID. Basically, you obliterate the precursor ion regardless of what the charge state is. When a beam type CID is you're applying a acceleration voltage between two different electrodes, basically, and this then is going to be dependent, of course, on the charge, not just on the mass. So one has a dependency on mass and charge state, and the other one does not. And there's also get different fragment ions. So beam type CID, you have these low mass fragment ions, ammonium ions. You get things like TMT tag ions. But you basically don't have any B ions or very little B ions. And so this thing becomes very different to differentiate whether or not one would be better than another. We had kind of a simple experiment where that is where we basically did a somewhat analogous to a SWATH experiment where we did 10 mass charge wide windows. We did some where these ones were all done with a single injection. These ones were done with 2 injections. So there is some overlap between the 4 and 5 mass charge isolation windows. This is kind of this other extreme, which is, again, Dave Goodlett had done this specific method in 2009. So here's the different methods. Here's basically the cycle time. Resonance CID always takes longer than that of beam CID, so it always has a worse cycle time. And so if you started to say, look, 3 seconds is the worst that we're going to allow for basically cutoff, it basically leaves just a few methods down at the bottom that we could basically use for quantitative data in theory. The effect, as we pointed out earlier, on the precursor isolation window is quite huge. Again, it's this inverse linear relationship between that and the precursor isolation window. But there's very little effect on things like the cycle time. And in almost all cases, resonance CID gives slightly better results than HCD. It's just slower. And so if you think about those as being quantitative experiments, you'd consider things like these. And so you do then make a bit of a sacrifice in the number of things you can detect. But all in all, it's these methods end up being definitely acceptable. But if you're going to make these things in wide isolation method windows versus 2 injections for these narrow isolation windows, there's actually a significant difference in the number of things that can be detected in any one run. The other thing we wanted to assess was whether or not there was a difference between what charge state you set in doing beam CID. So if you activated everything at a given optimal charge state, because you have a wide isolation window, you don't know if the things in there are going to be singly charged, doubly charged, or triply charged. There's probably a combination of all 3 in there. So you have to activate it not only at a given voltage, but assuming that voltage for a given charge state. So if you told the mass spectrometer that everything here is singly charged versus triply charged, it seemed like in general the things at a higher charge state setting for the beam CID definitely gave a little bit better results than the lower charge states. But if you looked at how variable the intensities were between any different charge state, ended up being pretty huge. So we have now 3 different measurements, and here's basically the coefficient of variation between those 3. And so if you're looking at the variance between MSMS that's done on one charge state versus another, there's actually a pretty big variance. If you do resonance CID, it's basically very little difference. So it doesn't really matter what charge state you basically set the mass spectrometer at. It basically obliterates everything, and the variability between any of the different charge states basically the same, gives the same peak area ratio. Now, I'll point out that in most cases, we just set the beam CID at a given collision energy, but I want to bring this up that if we set everything as being optimized for doubly charged, it's probably going to be less optimal for singly and triply charged. So the next final thing is product dyne selectivity. So if we had the ability to collect data at 30,000 resolving power, we can scan at 10 hertz. We can fill then basically for 55 milliseconds. If we have a resolving power at 15,000, we can have a scan speed at 20 hertz, but then the maximum inject time is basically 20 milliseconds. So in this case, in almost all cases, going slower with more maximum inject time and a higher resolving power works. And so, of course, we have two things that are kind of hurting the faster scanning. One is that we have a lower maximum inject time, so less sensitivity, and also a lower resolving power. But in this case, we find that it's much better to run the instrument at a much more conservative 10 hertz and collect the data at a maximum inject time for 55 versus this resolving power of 30,000. But again, all these are fairly narrow isolation windows. But again, I'll also point out, too, that this is also fairly narrow mass range, and we're getting an enormous number of peptides that we can detect. And we're also detecting, with a larger maximum inject time, more low abundance peptides. So these things do span, this distribution spans to a much lower intensity when we use a larger maximum inject time. And this basically shows the percentage of the scans that are hitting the maximum inject time, which means that they're underfilling the Orbitrap. And with the maximum inject time at 55, it's pretty rare, and that with a maximum inject time of 20, it happens quite frequently. So in general, this is kind of a little bit of a summary. So if you're looking for the ability to detect peptides, precursor selectivity is more important than cycle time. Fragmentation method, resin CIT gives better results than beam CID. In general, though, we've stuck with beam CID because it can go between our Q exactives and our fusion type instruments more readily, and the difference is fairly small. And product dyne sensitivity and resolving power tends to be more important than actually, I think they're about equal for that and about maximum inject time. So basically, as long as the product dyne sensitivity as a resolving power is, in the case where you're filling the Orbitrap, then the resolving power is more important than maximum inject time. But if you're not filling the Orbitrap, then maximum inject time becomes more important. Most people sort of ask, where do we start as far as instrument methods, because there's a lot of variables. We actually had this Nature Protocols paper a couple years ago. It's a little outdated now, but I definitely think that this is a good starting point for people as far as where to get started with different instrument parameters. There's a lot of things that are kind of being updated and improved, but this is a good first kind of starting point that gets you started. Okay, in the last few minutes, I kind of want to hit home in this area, and this kind of brings us back a little bit on why I think of these things as a targeted assay and why I don't think about these things really as a big discovery experiment. So the main thing is that once we started doing data independent acquisition, we kind of just dropped all of the stuff that we had learned for doing targeted SRM for about 5 or 6 years prior. And so what I'll talk a little bit now is kind of improving data-independent assay design using lessons learned from targeted assay development. So one of the first things that we learned really early on was lots of people were using DDA results in order to pick their peptides for SRM. This was really bad, and I can't say this in kind of a small, simple way. You're often going to be picking a lot of the wrong peptides if you're picking the ones that you sample frequently by DDA. One of the simplest things that we started to do really early on, Daniela Tomazella started taking every protein that we wanted to target. She would actually start by targeting every single peptide that in a given gene product. We then started making every proteins recombinantly and started doing that. And so this basically shows in blue is the DIA peak area. When on the orange plot here is the number of spectrum counts essentially that are in PeptideAtlas for this protein, Apolipoprotein B100. This is a very large protein. But you'll notice that a very large number of these most intense peptides that we measure by DIA peak area, many of them are undersampled by DDA experiments. So this is something that we found with SRM. So we started to do this as well also for DIA. And I'll talk on Thursday more on how we sort of recommend kind of trying to get around DDA libraries for doing our data independent acquisition experiments. The other lesson we learned, and we kind of stumbled on this by accident, and that not all peptides are stable. So this, I like to tell this jokingly just because I know that the postdoc who I'd say this about is kind of okay with me kind of joking with him. But I say that we kind of stumbled on this because the postdoc was kind of lazy. So often what would happen is what he did is he set up a 96-well plate to measure 96 proteins by SRM, and he was measuring every single peptides. But turns out that the 96 peptide was being injected on like a Sunday morning or something like that. And and he knew that he could calculate, of course, when his plate would be done. And instead of just coming in on the Sunday and starting his assay generation, he's like, you know what, I'm just going to inject them all again. And so then what happened is we had basically 72 hours or 3 days between the first injection of the plate and the last injection, and then a replicate of doing it all a second time. And so what we found was that a lot of the peptides were basically the same intensity between the two different injections. Some changed hugely, like completely disappeared. And so some peptides are stable and some aren't. And this became basically almost a standard thing that we did in the lab for SRM assay development, is that every peptide that we were going to measure went through a set of stability testing before we'd actually go through this process. Now, it turns out that this is really easy to do by DIA because you can do this for every single peptide all at once. You basically take your matrix, you do a DIA experiment, and then you wait 3 days, and then you inject the same sample again, and then you look at the peak areas between the two. In fact, other people in the lab now have just started making many injections of the sample over multiple days, and then they just look at the variance. So any ones that have a high variance, they just drop from their assay design right off the start. There's no point in going any further with those peptides. They will not give you good data. They will not give you good results. So it's good to be able to flag them to start with. Just because you can detect them in your runs doesn't mean that you can get good quantitative data off of them. And so here's basically just kind of showing this with the ApoB. The other thing that we also learned is that you should, and this we learned from the clinical lab. So in the hospital at the University of Washington, Andy Hufnagel goes and he has an assay for many different proteins. But one thing that he's always advocated is you actually have to have a digestion condition that's optimized for your peptides that you're measuring. Some peptides kind of come up really quickly and kind of plateau. Other ones kind of slowly come up in intensity and kind of go up then a little bit further. Some come up fast and then kind of go a little bit again. But in general, you wouldn't want to be measuring a peptide like this with only a 4-hour digestion time. You want to make sure that you have a digestion time that's out a little bit longer where you've given a chance to plateau a little bit. So let's say you're trying to make an assay for plasma. One of the things that we would do is we would actually do a digestion time course and ensure that the peptides that we're measuring actually have kind of come and stabilized at a fairly common plateau in the experimental sample prep that we're using. So again, this is done with one DIA run for 20 mass of charge isolation per sample. This does not take a lot of instrument time to do beforehand. So in this case, we've just basically redone that, but instead of doing this with SRM, where we only had 3 different peptides in our assay, in this case, we can basically go and look at every peptide we want to that's detectable. And some peptides kind of come up and then go back down again. There's this multiple kinetics of things, being released from digestion versus degrading. Other ones kind of come up, and don't give very much attention. Some come up almost immediately and then stabilize. Some take a long time to be released over time. We can also start to kind of understand some of these kinetics, right? So, Brendan mentioned a little bit earlier that Tina had mentioned a little bit about ragged ends. So we weren't really sure why this peptide was taking so long to be released from digestion. Well, it's because the first peptide cleavage occurs really quickly. That's formed almost immediately, but then the second cleavage takes a much longer time. So to actually be able to monitor this peptide, you'd want to make sure you go out to a much further digestion time course. So one of the fourth things that we learned in doing SRM, and that is that we needed to check to make sure that every peptide was actually going to be quantitative. So people started saying, okay, well, we couldn't do dilution curves or stabilized labeled dilution curves for every single peptide we're measuring. So how could we do this all in one go? So the idea that the lab had kind of come up with, and that is that we should take some pooled reference sample matrix and make dilutions in it in some diluent sample matrix. And in a case of something like plasma, we use chicken plasma to dilute it. In a case of an organism like Saccharomyces cerevisiae or C. elegans, we actually make 15N labeled analogs of the organism. So we're basically taking every single peptide and making a dilution of it where we keep the amount of protein loaded on column the same, and we're keeping a matrix that's somewhat similar. I will point out that when we've gone to looking at the matrix effects in chicken plasma versus that in human plasma, there definitely is differences, but it's actually a pretty good starting point. So just to kind of illustrate this a slightly different way, we tend to do 6 dilution points. We tend to take human plasma with chicken plasma, and then by the time we get down here, it's almost all chicken plasma and a little bit of human plasma. And the main reason for doing this is that in proteomics, or for any quantitative analysis, we have a calibration curve that looks kind of like this. We have the lower limit of quant and the upper limit of quant. And in most cases, one of our quantitative samples is going to be below or lower limit of quant, and the other one may be above. And this means that whether or not we can say our measure is actually just quantitative or whether or not it's just differential. So if you actually have one of your low points is down here and the other one's up here, while you can say that the difference that you're seeing from the mass spec measurement is statistically different, you can't say that the fold difference you observed here is related to the amount of moles that are in column. You're going to underestimate the fold change that's observed. And when you start to look at some data from this LFQ bench from the Navarro paper, you'll start to see the evidence at the lower signal intensity. Some algorithms tend to give an underestimate of the peak area. That's because one of the points is below the lower limit of quant. So we do this for every single peptide. And here's just showing a case of doing this with MS stats. And so here is this individual peptide. This is being diluted in chicken plasma. And and MS stats is saying that right here at this point is where the lower limit of quant is. And down here further is where the lower limit of detection is. Here's just showing another case too. We're taking 2 different proteins and making, our wild type yeast that we're using and making dilutions in a 15N yeast. The final thing that we've done is that if people, and this is something we started to do when we started doing targeted proteomics that span multiple different days. I'm nearly done, and that is that we needed to demonstrate that we could make what was the within day and our between day precision. So this is something that Andy Hoofnagel and Russell Grant kind of pushed us on to doing. And that is that we start by taking 25 samples, we stick them in the freezer. In day 1, we take 5 out, we digest them and run them. Day 2, we take 5 out, digest them and run them. Day 3, we take 5 out, digest and run them. We can use this then to assess our precision and our measurement. This does require a decent amount of instrument time, but it's actually definitely a very worthwhile in assessing your within day and between day experiment. You could even narrow this down to like a 4x4 or a 3x5, etc. It wouldn't be too hard. And the other final thing is that don't use a large library just because you can. So this is something that everybody wants to see. I want every possible peptide I possibly can get in my samples. So they'll even go as far as taking samples from some neuronal cell line and searching it against, searching their HeLa samples against it, just to see there may possibly be some peptides that are specific to that neuronal cell line that they may find in their HeLa sample. In general, you want to search with a much smaller, you only want to search for things that are likely to be present in your sample. And everybody talks about the occasional times you get the odd few peptides because of that, but people don't really talk about the things that they lose because of that. And so let's just assume you have here an incorrect PSMs and your correct peptide spectrum matches, or just in case you have, in this case it's peptides. And if you set a given 0.1% FDR threshold, and let's say you took a much, much bigger library of only which a small portion were actually going to be in your data set, you're actually going to be increasing this to a much greater degree than you will be this greater file, this distribution. So then you have to shift your threshold further over to the right. This then makes it less likely to be able to detect any one of the peptides that you could have detected with a smaller assay library. So again, this is actually a fairly simple to do. We do this now on 67 LCMS runs. This isn't too routine yet, but it's becoming more routine in our lab, and it's something that we've definitely learned every time we've done these experiments. So just a brief summary here is that Orbitrap mass spectrometers are a growing hardware solution for performing data-dependent acquisition. I'm actually impressed with seeing how many people are starting off with thinking about this on Orbitraps. Tools for the analysis of data, DIA data fall into two main categories. You'll hear from Alexi on spectrum-centric workflow and peptide-centric workflow. We definitely think people should be thinking about both depending on the applications and the needs. DIA data collection requires balancing different parameters. So narrow windows are important for selectivity. Filling the Orbitrap is important for sensitivity. Both narrow windows and long ion times reduce the comprehensiveness of the assay. And we should not forget the assay validation lessons we've learned from targeted proteomics in the past. And it definitely is something that we continue to strive for. It's no point querying for peptides that are not going to be quantitative to start with. So if you have peptides that have poor precision, aren't stable, have bad digestion kinetics or bad linearity, you might as well drop them from your assay library. And that will actually increase the sensitivity of the peptides you can actually detect otherwise. And thank you very much for your attention. I'm happy to answer any questions.
Great. Thanks, Mike. Maybe I can sneak in a quick one before we start.
Okay.
I'm interested in your LOD, LoQ slide between MS1 and the different windows. You don't need to go back, but I mean, so we also see this strongly more sensitivity in MS2, and we also see when we shrink the windows that we get better selectivity and you identify more peptides. So I think I'm interested in the difference between the instruments. So on a QTOF, this can be explained mostly in terms of selectivity, and you also pointed to selectivity. Yeah. The question, and we also talked a bit about this yesterday. So is there, on the Orbitrap, do you think there's a strong effect of, because you're shrinking the window, the fraction of a given target analyte is more of the ion current that you're getting? Is there a strong effect of this as well? Or? Yeah,
so a huge component here is automatic gain control. So what people don't realize is that when you're filling with 1 million ions in an MS1 scan, you have 1 million ions before you hit space charge effects on an Orbitrap. You've got 1,000 different molecular species in in a very large mass range. Let's say 1,000 mass to charge. When you now fill for over 10 mass to charge window, you're now filling 1 million ions in a 10 mass to charge window. You're effectively enriching the time in that narrower window. And so that means that now you're only increasing amount until you hit that maximum inject time, which is why we think keeping the maximum inject time high is a pretty important component, because you want to basically ensure that you always fill the Orbitrap, which means you gain the highest enrichment that you possibly could.
So with these brighter sources, this is going to mean your maximum inject time can go down a little bit.
Yeah, that's definitely a case. So we'll talk a little bit more on Thursday about this experiment we did, and we published in 2013, where we multiplexed multiple different fills in the Orbitrap, and it really helps for selectivity, but it really killed us as far as sensitivity goes. We now feel like with the Lumos and the QE HFX should have the exact same advantage, which is a brand new Q-Exactive instrument that's just released, that you basically can get by with 15 to 20 millisecond fill times in some cases.
Okay. Other questions for Mike? Timo?
I have a question on your spectral library generation that you just mentioned in one of the last slides. So do you have any numbers in some way? So where is a critical, let's say, threshold? It's not really strict threshold, I guess, where you blow up the library in a way that you have negative effects.
Yeah.
And what would you recommend with respect to generating libraries on some cell sources that you would like to feed in? So something very specifically for a project, if you have several, let's say, stimulatory conditions of your cell line of interest, or different cell lines that you would like to compare. So a smaller set, but equally distributed of these cell lines or cell states that you're interested in order to make a library, or what is a recommended setting?
This is kind of a really long answer to this, and so I wonder if we can get to it to Thursday, I think. So on Thursday, I think I'll point out that I don't think DDA is the best way to generate a library. And there is a simple way of just generating a DIA library from DIA data from just about 6 injections on your actual samples. And so that gives you a lot of power on your actual samples, on your column at the exact same day, on the same instrument, and it takes just 6 injections. And there now we are able to detect about 90% of the things that are in the library. We're able to detect in the wide window runs that we do later. But that's kind of something that's all unpublished. It's all very new. So I felt it fit better under what Ben had suggested as being kind of next generation new ideas on Thursday. So I think, but that's a really good question. It's hard to, I just wanted to sort of just point out the fact that people have always thought the bigger the library I get, the better the results I'm gonna have, and I don't think that that's necessarily true, and people just have to be cautious with that. Sometimes it may be better to have a sample-specific library that I think would lead into that a little bit better. But I'll talk about how we do it specifically on Thursday.
Can I just jump in with one comment? Is that that what he's going to talk about on Thursday is sort of just one case of what I think I learned at one of these courses, or just sort of realized at one of these courses, is that there can be 2 phases, and you can use DDA, you can use DIA, you can do runs, like runs separated by 36 hours to look for degradation. You can do a set of runs on pooled sample and use that so you could start with a huge library, which we frequently do, even in the cases that Mike is talking about. We would start with something like a pan-human library or something really big, but then rather than immediately taking it to quantitative runs, we would do a set of, of pooled sample runs, and those might be gas phase fractionation, or they might be, like different digestions, or leaving in the autosampler. You could do all sorts of things, and then you can filter on what you can detect. You could filter based on CVs. Even if you do half your runs at different times, do you still get a less than 20% CV over time? And you get a filtered library, and then you get to take that to your quantitative tests.
In fact, it fits really nicely into what we used to always call in our SRM assay belt of refinement. So we have like kind of this refinement step of which peptides are stable, which ones are precise, which ones have good retention time characteristics. And so so what Brendan's bringing up is basically we're using the same analogous strategy in order to do things in our DIA data. And I think I'll tell you some very simple ways in which we do this on Thursday, which I think I'm pretty excited about them because they make the workflows a little bit easier.
Tina?
So you show these nice figures with highlighting that different peptides are differently digested in time. So from what you show, it looks like that it's always pretty much beneficial to do digestion overnight, or to do it rather longer. Yeah. Or do you also have experiences that it actually is not good to do it too long? So what is your recommendation there?
So in our lab, we kind of settled for a while on 3 to 4 hour digestions for a long time in the lab. And that was because we just got more DDA IDs at that point. And so because of that, we just stopped. And we didn't think about whether or not this actually gave us good quantitative data, because all we were focusing on is a bigger list at that point in time. Once we started looking at the fact that when we were doing these things much more in a targeted type analysis, we did find that longer digestions were better. We also found that adding a much higher concentration of trypsin helps in a lot of these samples. So in cases like plasma, you have to just basically shovel in trypsin because there's a lot of endogenous protease inhibitors. So we often add a 1 to 10 as opposed to a 1 to 50 that people often use of enzyme to substrate ratio, which is, we learned that from those digestion kinetics. And then we were able to get those curves to come back up again into the 5 hour type range. So part of the reason for the short time scale is that the clinical labs don't want to do overnight digestions. They would rather do a 1 to 2 hour digestion and then run samples and they get data back to the patients again. And so because we have a couple of assays in the lab, which we are thinking about moving over to the hospital clinical lab, that forced us to think about things as a fairly quick turnover. So, but we wanted to make sure the peptides that we were using were at least stabilized at the time point we were using. But that's a good observation.